So what I'd like to do then is to segue that just into a question to kind of parameterize your sense about how this would be applied to a real kind of analysis. Let's say we're looking for single letter changes. You may recall that these are sometimes referred to, the technical term is single nucleotide polymorphism. It just means one particular position in the genome where one letter is changed in some people compared with other people. So some people have an A there, other people have a C there. And let's just say, for the purpose of argument, we're looking for one of those that's pretty rare. Maybe only 1% of the people have C and everybody else has A. Okay? So let's say we actually go out and we collect DNA from a bunch of people. So we have a different DNA sample from each person. And now let's say we take each one of those and run it through this kind of short read sequence. So we're going to get a big data set for each person. Now, of course, the machines themselves are imperfect. The fact that they can read DNA at all is kind of amazing, but they make errors. So a typical kind of error rate for this sort of process is about a 1% miscall rate. That means any given short read that you've got, on average, one of those letters out of 100 will be read wrong. Instead of reading A, it says T, or it says G, or something like that, okay? So the question is, if we do this analysis, will we be able to distinguish a real case where a particular person actually has the polymorphism, in other words, a C instead of an A, versus background sequencing error? So, just to make the obvious point, the numbers here are the same. Rare polymorphism present in 1% of people versus a sequencing error background miscall rate of 1%. So let's say, again, for the purpose of simplicity, our coverage in each one of these DNA sample sequencing data sets is on average 50x coverage. Okay? Any questions about what I mean in any of this? What the words mean? Okay, so basically what you're being asked here is, do you believe that this kind of data set will provide us a basis for reliably being able to distinguish the people who actually have this polymorphism versus the possibility that what we're seeing is just due to background random sequencing error? And as a sort of a sub-question that sort of asks you what you expect to see under those two different scenarios that we're trying to distinguish, if I say we have 50x coverage, that means we're going to have 50 reads lining up to that particular letter where we're looking for this known polymorphism. So what would you expect to see in those 50 reads under the two scenarios? One, that there's a real polymorphism present versus the sequencing error. And of course, that's intimately connected to the question of whether you think those two scenarios are reliably distinguishable or not. Any questions about what you're being asked? Yeah, let's assume, it's a known polymorphism, A versus C, and it's at this particular position, and we've got all the reads all properly aligned to the genome. Let's assume that we're not going to make any errors in aligning the reads. Everything's properly aligned, so you know exactly what reads align to that position in the genome. You know what you're looking for. Let's say, A versus C.
Are we aligning it to the genome without the single nucleotide polymorphism?
the alignment quality basically doesn't really care at the level of like a single nucleotide difference. It'll accurately align perfectly fine whether if there's a one letter difference out of 100.
So this SNP is found in 1% of people, so that's a single nucleotide that's different from the whole.
Yeah. Let's say that 99% of people have an A at that position versus 1% of people have a C at that position.
But each person has many of these short reads, right?
So now you're getting into the underlying model of how the data is going to be derived from the person, right? So all I said here was that you get a DNA sample from each person, and you do short read sequencing on each one of those samples so that you have a separate data set, one for each person. And in each one of those data sets, you have 50x coverage on average across your whole genome. Okay. Well, why don't you take a minute to think about it. You should see the question on your laptop or smartphone. You seeing the question on there? Okay. So just think about it for a minute, and then enter a brief answer.